At this time, we will hear from one of the three winners of the Texas A&M Graduate Student Oral Competition. 3 students studying plant breeding and related sciences were selected among a pool of applicants to deliver oral presentations during the symposium. Mitchell Schumann, an A&M graduate student, was selected to present during the symposium due to the relatability of the research to our theme. Mitchell Schumann received his bachelor's degree in plant and environmental soil science from Texas A&M University in 2012. He then attended North Carolina State University, where he worked on association mapping in autotetraploid potatoes, and then graduated with his master's degree in 2015. He returned to Texas A&M that same year as a Cotton Incorporated Fellow, where he currently works under Dr. Wayne Smith towards his PhD in plant breeding. The title of his presentation is Identifying Potential Nesting of Fiber Quality Alleles Within the Population Structure of Obsolete U.S. Cotton Cultivars. Please join me in welcoming Mitchell Schumann.
Howdy, Ags. Howdy, honored guests. Hope you don't mind if I stay down here. I'm a pacer by nature. I'm afraid if I get on that little stage, I'll just fall right off. So anyway, I got a lot to talk about, so let's go ahead and get started. I'm glad I get to add an element of diversity to this diversity symposium by talking about a crop other than a food crop, and that's cotton. Cotton's a very important crop. A lot of us are wearing cotton right now. I would be a lot chillier if it wasn't for cotton right now. The U.S. is the largest exporter of cotton in the United States, or sorry, in the world. And Texas is the largest producer of cotton in the United States, so it's very important to our economy. I was listening to a kind of a State of the Union address from Cotton Incorporated, and one of the things they mentioned was producers are looking for fiber quality as one of the most important things they're looking for from science. So I believe that's probably part of my funding here through the Cotton Incorporated Fellowship. So one of the reasons this is important is we sell a lot of our cotton to overseas. They use spinning technologies, and these spinning technologies work best with higher fiber quality. And of course, it allows them to produce better products, and it allows the farmers to obtain a premium as a result for these products if they can get these fiber quality traits into their package. So there are 2 primary methods of phenotyping fiber quality, and that is HVI. HVI has been around since the 1980s, or since the 1960s. It began broad implementation into selection schemes and breeding programs in the 1980s into the 1990s, and pretty much everybody uses it now. Advanced Fiber Information Systems, which is APHIS, was developed later. The difference between these 2 methods is HVI looks at bundle characteristics for fiber quality, and APHIS looks at individual fiber. They're both really good systems. APHIS isn't as broadly implemented because it is more expensive, and it hasn't quite reached the full level of acceptance in the community yet. So let's talk a little bit about cotton genetic diversity. Majority of all cotton in the United States can be traced back to a single introduction in 1806. And there's stories that was actually smuggled in from Mexico, stuffed in dolls. That's how we got that seed here. So we have a very narrow germplasm base to begin with in the United States. There's many genetic studies that have come out that kind of testify to this. Now, one of the good things that came out of my old alma mater, North Carolina State, is there was a diversity analysis that said, well, because of this narrow germplasm diversity, we might be able to tap into traits of interest in the obsolete germplasm collections of the USDA's germplasm collection without taking as much of a yield drag. So that kind of started my question is, can we tap into alleles that may have been overlooked from the USDA obsolete germplasm collection using genomic selection? So in order to start that question, you have to look at population structure of the USDA germplasm collection, and there has been analysis that I've been able to work with to begin that process. So the first thing we should talk about is what I'm working with. I worked with genomic information from 258 U.S. obsolete cotton cultivars. That is a subset of the U.S. obsolete germplasm collection, and these were selected to represent that collection based on an SSR study that categorized the populations, the subpopulations within the collection. So what I did is I took that 258 lines and I genotyped them using the 63K SNP array. A lot of this work is actually shared. Some of these lines were already genotyped by Dr. Stelli's lab and Dr. Laura Anzi's lab, and we did a subset of that as well. So after I did all the filtering steps on that array, we wound up with 23,000 SNPs. I have phenotype data on 128, which is a subpopulation of that 258, because it's a lot of work to phenotype. So we did 3 replications at two locations in RCBD. We originally had 3 locations, but after getting washed out 3 times, I admitted defeat. So we phenotyped this using HVI and APHIS. And we are waiting on spin data. Spin data is very expensive, and it takes a lot of cotton to develop that data. So we can only do spin data on 1 rep at 1 location. But what I'm going to do is use the HVI and APHIS to predict what parameters are best for predicting spin quality. And then I will predict the genomic parameters to those HVI and APHIS data. So when I first started, I said, okay, let's look at structure from a classical standpoint of where these lines were developed. And we have 4 distinct growing regions of cotton in the United States: the far west, the southwest, the mid-south, the southeast. So first thing I did is I said, okay, let's run a principal coordinate analysis and just color it by these locations. And as you can see, it's pretty, but it's not very informative. So one of the things I did to kind of put a little bit of a control into this study is I inserted these TAMU lines. And a lot of these come from the same parental sources. So I expected distinct structure within these lines. And you can see they kind of formed a tight little knot down there. But then this isn't really informative just looking at location data. So I said, let's infer population structure using the marker data itself on all the lines. So I used, well, actually, first you can see why a lot of those locations were intercolored. This is a cluster diagram separated by location. I went in and actually looked at the parental history of these. And I found that some of the parents actually went across many of the lines for geography. So that's why we saw all that different coloration right here. So anyway, to look at the infer population structure from the markers themselves, I used fast structure. It's basically what was formerly structure. It's just a more efficient algorithm for using it. I still had to link into the supercomputer to do this because it's very computationally intensive. So when you run structure, you estimate how many subpopulations there are to begin with, and you do an array throughout that of all different possible amounts of subpopulations. So I chose k equals 1 subpopulation through k equals 10, and I ran it through that array and looked at and compared the models, and I found that the output of that model said that somewhere between K1 and K10 subpopulations is the best amount of populations. So awesome. It wasn't very informative, but I said, you know, maybe we can combine this with PCOA and make some sense of this information. So if you remember, I inserted that control population of those TAMU lines, and I found that at k equals 4, I was able to get that population teased out, as well as have good separation between the colors, for the most part, between the other subpopulations categorized in this study. So what I wanted to do next is I want to see, hey, can I associate the markers with population structure? What markers are informative in predicting population structure? So the first thing I did is I took all 2,400 markers and I did a single marker analysis using logistic regression. I looked at all those coefficients. I performed a walled test on them to get a p-value. Obviously, I have more predictors than observations, so I had to do some sort of false discovery test. I did false discovery rate analysis. I found 2,800 SNPs that were associated with population structure. I found it is best to look at structure through a multivariate analysis using all the markers simultaneously. So I wanted to use some sort of multivariate test. I used random forest because I like the feature selection process of that, and I also like the robustness of random forest in general. So the first thing you got to do when you do random forest, you got to optimize the tuning parameters. and then you run the model on those optimized tuning parameters, and then you can perform feature selection. For those who know about random forest, I just did this by using the out-of-bag error rates. I took out all the features or markers that were not important in predicting the model, and then I ran it again. I actually programmed this into a loop in R, and I just let it run as many times as it could until I finally weeded out all the markers that were not informative in predicting population structure. When that was said and done, I had 1,200 SNPs that I found that were pretty predictive of population structure. And just to show that these markers that I pulled out weren't necessarily informative, I ran the model with the full set of markers, all 24,000, or sorry, all 2,800, and then I ran it with the reduced set, the 1,200 that were left after weeding out, and I performed K equals 10 folds cross-validation, and I replicated that 10 times because I like to burn up processors. And I found that if you look at the array of predictions for, or prediction error, there's really no difference. So the markers I pulled out, I'm assuming, are not very informative in population structure. So from then, I took those 1,200 SNPs and I just did a simple single marker analysis using a linear model. And again, I used false discovery rate correction, and I found 416 SNPs that were nested within the population structure that seemed to show associations with the trait of interest. There's many traits in fiber quality. For this presentation, I just chose length. So, all right. So the next thing I wanted to know is, we have sequenced the cotton genome. So we know the positions of a lot of these SNPs in the genome. So I went ahead and mapped them. And I found that a lot of these markers co-locate to for pretty much the same areas. Remember, they're nested within structure, so I kind of expected that to some extent. So if you look at the red lines, this is where their location is, but within each of those red lines, there's several markers that co-locate to those spots. So the next thing I started to wonder is, okay, because there is population structure, in some of these clusters, I'm assuming that there's no allelic variation within the markers in this cluster compared to maybe markers in another cluster, which might have, or markers in another subpopulation that might have a lot of allelic variation. And it turned out to be the truth. And so what you're looking at here is I colored the locations on the genome that were nested within the population structure that clustered together and had the most allelic variation for the exact same class. So what you're looking at on the one, say, right there in the middle, is subpopulation 4 had the most allelic variation for all these markers which are located in that region. And it was interesting, if you look at, I think it was subpopulation 1 in that, for all those markers, they were fixed. So it seems like it allowed me to identify these traits of interest nested within the population structure. So how can we apply this? Again, there are some regions that are fixed and some regions that there's a lot of allelic variation. So maybe it would be best to keep these populations separate and breed them within those regions first, and then maybe use marker integration later to bring the populations together to get all the alleles that are of interest for our breeding program. Of course, we are living in a modern genomics era, so there's other modern genomic techniques, biological techniques such as maybe chromosome substitution or even CRISPR-Cas9, where you can instill allelic variation in regions of interest that we found if we want to stick within one population where it's fixed in that region. So where does the genomic selection apply? We can still do the model by creating a training population that emulates the obsolete cultivar collection. And this was a proof of concept paper in Nature that showed that you can do that. And, but my question is, what if we took these markers out of the genomic selection model, the markers that are associated with the trait and predict population structure? Maybe we won't be selecting with a bias. I need to look into this more. I'm looking at ways of synthesizing a population in silico and analyzing it to see if that's true or not. One thing I'd be remiss if I didn't point out is the best policy in doing these type of analysis is to have every single step in a cross-validation loop. I did not have I used different softwares, I used supercomputers, I used my own laptop. It was very unpractical to put all this in a cross-validation loop. I tried to take measures for protection. I used a population that had that was a control population with those TAMU lines, and I expected certain results of that, and I got those results. Also, I did use cross-validation in my random forest and my multivariate modeling, so I did what I could, but really you want all this in a cross-validation loop to avoid overfitting. So in summary, it seems like this analysis does work. It's going to take more testing, of course, as with every presentation. This information could be useful in developing training populations for genomic selection, as well as developing GWAS populations in the future, and in just basic general breeding decisions. So there is a potential problem, though, for overfitting with what I did. So I want to say thanks to all these people, and I guess I got to go.
Thank you, Mitchell.
Do I have time for questions?
We can take one question.
Yes.
So I'm wondering, the fact that you had so much difficulty finding structure, that maybe that is good news.
That was the initial
because you
assumption.
can take the whole domesticated cotton germplasm of these 4 regions, treat them as one population, and do GWAS if all of them get phenotyped for whatever trait or quality and so on, actually. Isn't that the way to go?
Yes, we considered that. That was one of the original thoughts, is because there is such a narrow germplasm base. That's why we think we can select across lines. However, we do see that there is some structure elements when we bring lines from the southeast, and they don't perform very well in fiber quality. They perform much better for fiber quality on the East Coast. So we know that inside the genetics there is some structure issues, even though we're not able to easily identify them in a structure analysis.
But maybe that's precisely having like a common nursery planted in these 4 regions would allow you to maybe better, and with an increased number of markers, better identify which of maybe a fairly small region is actually responsible for these differences.
Yes,
I'm not sure if that exists in cotton.
that would be great if we could. I don't know. I guess we would have to put the logistics together of a big collaborative project with multiple locations for growing.
Yeah.
But that would be a way to go. Yeah, absolutely.
That's all the time we have for the questions. Let's thank Mitchell one more time for his presentation.
Thank you.
Now we will be